Hello and welcome to the NSolver Analysis Software webinar series. The series is comprised of 4 short training demonstrations on how to use the NSolver analysis system. The quick start is a very brief end-to-end demonstration of how to use NSolver at a high level. The other 3 webinars are short videos, each focused on one of the 3 main components of NSolver: import and QC, analysis, and finally export. While these 4 webinars are general to all assay types, they are focused particularly on gene expression and microRNA. Coming soon in our next series will be data type specific videos on copy number variation and plex squared assay types. This webinar will demonstrate the export and integration capabilities of the nCounter data in the NSolver system. NSolver is a desktop solution available for PC and Mac compatible computers. It allows you to view, sort and filter your data in a table-based format. You can quickly analyze your data from your past experiments, and you can analyze fast using NanoString's recommended protocols, or you can customize those analysis methods. And full analysis is accomplished in 3 steps as shown below. The import wizard allows you to import the data and carry out QC. The experiment wizard allows you to normalize the data based on housekeeping genes and build ratios. And then the export wizard allows you to export the data and also to integrate it with other external software programs. And today we're going to demonstrate the export wizard. So without further ado, we'll move into the demonstration. Now let us look at exporting data. So throughout the application, you can export data in several file formats for use in other applications or analysis tools. NanoString has formed partnerships with several software visualization and analysis companies. Some partners, such as GeneData's Analyst, support direct import of the RCC files. Other exports are available directly from the NSolver system, and I'll show you them as we go through the system. For copy number variation users, the NSolver system exports in a format directly compatible with BioDiscovery's Nexus copy number. This will be discussed in a later webinar. It's very important to realize that the order of rows in the viewer will be consistent with the order of columns when opening a table as well as exporting. To view a table or export with columns in a specific order, sort the rows as described prior to exporting or launching the table. The ratio description field in the experiment wizard can be used to assist in sorting of ratio data in tables and columns for export. We can export all the data. So for instance, if we wanted to export all the normalized data without any filtering or anything, we can just select it all and click export. And we'll now be presented with the export window, which I will take you through in a few minutes. Another important feature of sorting that needs to be recognized is that any sorting that's carried out on this main table will be carried forward when you export or when you open a table. But any sorting that's carried out on a table is not going to be carried out when you do the export. So it's important to do your sorting on the main window so that that information is still in the same order that you wished. Remember, each of these samples will be sorted and there'll be a series of columns along the top in the table. More normally, what many people want to do is export a subset of the data. So I'll give an example here using the ratio data from this experiment. So if we look at all the data in the table format, because we want to export a subset, if we wanted to export all of this, we could have just done that from the main window. What we're going to do is filter it so that we only see those genes of interest with at least a 2-fold absolute fold change. So we can filter by choosing our comparison, the juvenile versus adult. You'll notice that when you change the filtering, the matching options will change to be relevant to that type of data. This is numerical data. So let's look for all of those first that are greater than 2. As you can see, what we're seeing now is all of the data where we have greater than a 2-fold change. But obviously, most of the time, people want to actually look at things where you've got an absolute 2-fold change. So let's actually look also at those that have been decreased by 2-fold relative to our reference batch. So now we want to do OR, and now we will say those ones that are less than -2. So now we're going to see all data between 2 and -2. Above 2 or lower than -2. So absolute fold change of at least 2. And now if we say go, we can see all of the data. We can sort it so that we have it in whichever order we would like. And then we are ready to export. The reset button allows you to reset your filtering and go back to the basics at any time. It's also possible to export specific genes for export within a data table without using the filter function. Simply select the genes you're interested in and click the export button. The export wizard then allows you to export of only the selected genes to a text file or other application. Alternatively, you may only select items of interest for export, right-click and choose Save. This will allow for saving the selected items as a CSV file and will not launch the export wizard. And as with all of our exports, you're able to open it as... So that was that selected export. Here I've selected the data that has an absolute twofold variation. And now we wish to export it. So we click on the export button and we're offered a variety of export options. Custom export is the most widely used export option due to its flexibility. Exporting using this option allows for direct import into several visualization packages such as GeneCluster Java Tree View, which is open source, MEV, which is also open source, and FDA ArrayTrack, which is open source, as well as GeneSpring and the Rosetta Resolver system. To export using the ENSOLVER custom export, choose the custom text format export. To automatically exclude from export all data that was flagged in QC or normalization, click on the checkbox under Common Options. You can now customize the format and columns for your exported data. Click on the checkbox next to only the columns that you require to export. In addition, you can choose the header you want to display for the data columns. Choosing to export the ratio data description will overwrite the default ratio numerator in the exported file with the description that was entered on the final screen of the experiment wizard, or you can use both. If I were exporting raw or normalized data, I'd have the option here to export description, which would replace the RCC file name with the description for the file that I reviewed in the import webinar. Likewise, for raw and normalized data, select Export Group Names in order to export the annotation group names. To export all data in a single file, select Single File under the split options. This is the most common use. To export each sample lane separately, choose the 1 ratio per file, depending on what level of data you are exporting. Columns for export can be customized. Files failing QC can be automatically excluded. And for ratio data, data can be output as either fold changes, log2 ratio, or ratios. You can also choose whether to export the numerator and the denominator. And you can choose to export data as either a CSV file or a text tab delimited file. We click Finish. We're asked where we want to save this. And when we've actually selected a location to save, we're also asked if we'd like to open the file immediately. And here we see an Excel file containing the data that we chose to export. The RCC collector format. For those that are familiar and prefer the RCC collector tool export, this option is available for all raw and normalized data. So if we select some normalized data and we choose export, you'll see that the RCC format option is available. This combines the individual RCC files produced by the nCounter Digital Analyzer into a single spreadsheet for analysis. It also includes specific header information that's different than the custom text format export option. As you can see here, this is the header information that is different from normal. ENSOLVER contains seamless integration with the Ingenuity Pathway Analysis software. The IPA system is a web-based application used by biologists to better understand the biology underlying their experimental results. Scientists can concurrently analyze multiple data sets across different experimental platforms and identify key functions and pathways that distinguish biological states. This technology is especially useful for dose-response assessment, time course experiments, and simultaneous analysis of gene lists and protein lists. The web-delivered application makes use of the Ingenuity Pathways knowledge base, a database consisting of millions of individually modelled relationships between proteins, genes, complexes, cells, tissues, drugs, and disease. If you have an Ingenuity license, gene sets of interest can be uploaded with their associated fold change values to the IPA system for further analysis and investigation. To upload data to Ingenuity using the NSolver system, first select the data to be uploaded as we showed before, and then choose Upload Set to Ingenuity. Select the Upload Multiset to Ingenuity. If you want to exclude lanes and samples that were flagged in the normalization, click the Common Options box and then click Next. Enter a unique project name and dataset name. Choose the correct gene column name and gene ID type. Typically for nCounter data, this would be the accession number, the RefSeq ID for gene expression experiments, and the gene name MirBase for microRNA. Then click Finish. And then you'll be automatically launched into the Ingenuity application. You can also export in the Partek Genomics Suite format. Partek's Genomics Suite provides support for both raw RCC data files as well as NSolver exported data files. NanoString recommends importing your data into NSolver, creating an experiment, and then exporting the normalized data using the customized Partek Genomics Suite export. So if we select our normalized data, we select what we want to export, choose the export option, select the Partek's Genomics Suite export, and we can exclude the lanes or samples that were flagged in normalization if we wish, and then we can export it. It's exported to a CSV file that you will import into the Partek system. And here is the resultant file. If you look at the data file, you'll notice that the Partek Genomics Suite export format is inverted with respect to other NSolver exports. The genes are displayed in columns rather than in rows as in other formats. For information on importing your data into Partek Genomics Suite, call your FAS or your Partek representative, and any of us will be able to help you. Finally, it's important to note that NanoString provides support for the export function only. Interpretation of data outputs for all partner software packages are up to individual users. For information on purchasing or details on partner software packages, contact the appropriate company directly. And the final thing I'd like to demonstrate is the experimental report. These are available for all experiments that were built using the Experiment Wizard. These reports contain information that tell you all the parameters you used on the QC and analysis of your experiment. In addition, it provides some additional QC statistics on the raw data and can often be very helpful. To view the report, select the experiment, so in this case the experiment is sample set 1, and click on the report icon. The report will open in HTML format and contains basically all of the underlying information. Your name of your experiment, the raw data you used, the results of the QC experiments, the values for QC experiments, what type of normalization you used, the positive control normalization, the code set, so again this is a good way of seeing which housekeeping genes you used, the ratio data you did, the results of the positive controls and the negative controls. And this can obviously be saved or exported using the appropriate controls in your browser. So thank you very much for listening. As always, further information can be obtained from the NSolver user manual, which is available for download from our website or was actually in the NSolver installation zip file that was downloaded when you installed NSolver. If you have any NSolver specific questions, please contact NSolver support. Any general nCounter questions, please contact our support at nanostring.com. And if you wish to purchase any nCounter products, please contact sales@nanostring.com. So I'd like to thank you very much for listening to this webinar. And soon we'll be presenting some webinars on CNV analysis, Plex squared analysis, and also advanced analysis. Thank you very much.